Indeed, we have begun. So, we're going to pick up where we left off on the last time we were together. Let me get the housekeeping stuff out of the way. So, I realize you may have other classes, and I said to you before, just to reinforce the point, as soon as you get here on Wednesday, you can grab an exam and start. Again, they're going to be 3 different colors. They're all the same exam. I'll have to hand the exams out depending on where you're sitting, just because there's such an overabundance of people. In all the classes, we're basically sitting on top of each other a lot. So even if they're, I'm sure some people will be done and we'll have room in here. And if for whatever reason you need an extra 5 minutes or so, because I will want to be out of this class at 10:50. There's another class coming in. Just go right over next door to the lab. It'll be just fine. There won't be anybody in there unless Dr. Robbins may have some stuff set up. But he doesn't start his lab until 3 o'clock on Wednesday. So you can take your stuff and migrate over there for an extra 5 minutes or so, if necessary. So I don't want you to feel that time constraint. So I'm roughly allotting an hour or so for that. But I'm trying to be as flexible as I can given the constraints that we have here. That's number one. Number two, nothing on your desks when you take that. Put all your stuff under, leave it in the book bags, whatever. No cell phones, no nothing. That's a bad look, and that can have implications. So we don't want the appearance of impropriety or impropriety is something we wish to avoid at all times. So I'm not making accusations. I just don't want anybody to be upset about that. And again, the last part to read, I didn't even reinforce it with the question. If you have one of these things with the ADA where you can take it in a different environment, that's an arrangement you must make on your own if you choose to. I have nothing to do with that. I only work with the people there if someone has made that appointment. Very good so far. We are good to go. So I'll finish up today. There's not a whole lot with this. This is about the nucleus. You've heard all this stuff before. That's there. Let me just get to the right click. There it is. I've clicked. For our purposes right now, we're not worried about. We're just looking at the uninucleate cell. In general, cells that are multinucleated have more advanced functions. The liver is sort of an oddball in that. And down the road, just red blood cells shed their nucleus because they don't need it anymore. That's really a sign of the advancements they have and what they've become, technically. So none of that is a big deal. Yeah, we know what it does. The components are such, there's not a lot to say about the components per se. It is a double membrane. That's notable. And I guess the one way to look at it is the outer membrane is sort of the beginning of what we call the endomembrane system. I talked about the last time we were here, the series of membranes that protect materials, not the least of which are things like RNA, messenger RNA, transfer RNA, ribosomal RNA, other materials that are either coming in or going out of the nucleus. Again, the DNA can't get through. It's too large. There's an abundance of pores. The inner membrane is kind of like what we see on a lot of the cells, sort of giving it a certain structure. As you'll see, they describe it. And then the nucleolus, you'll see that everybody has the same opinion about the nucleolus. That's where ribosomal RNA is most likely made. That's sort of a consensus type of thing. It's on the next test. I've never given a test about parts of the cell without using that particular question. What is the presumption of the activity of the nucleolus or nucleolus? It makes ribosomal RNA. See, everybody's guaranteed to get one right next time. And then this stuff, it's already an error. Chromatin is not condensed here. This is the uncondensed form of chromatin. What happens is chromatin really represents a combination of DNA and proteins mostly. DNA's natural tendency is to supercoil. It's like a spring. So a tiny spring is the best evidence of what DNA is. Okay, what happens in chromatin, we have proteins called histones, Histones, that you will see, that basically it's as though you took them and inserted them in between the little rungs of the spring. And it doesn't allow it to really spring. It just kind of stretches it out. So You'll see it in a second. And a lot of work has gone into studying histone proteins because what happens when the histone proteins magically disassociate from the DNA strands, it supercoils, which means it's ready to undergo mitosis. So they've been studied as a way for controlling mitosis. That's really the thrust of today's lecture, is to understand why mitosis is so important, because mitosis that's out of control is what cancer comes from. It's all about the cell cycle losing things that allow it to be maintained under control. This is really the frame of this particular presentation today, is to understand something that's universal about the body. We tend to do things too fast. Our heart rate, without the controls we have, would be over 100. If you had a resting heart rate of over 100, that's a burden on your system that you don't need. So we have a system that we learn about at the very end of this year called the autonomic nervous system that governs, holds back our heart rate for most of us between 60 and 80, which is normal. And it's a very good way of assessing your health. It's a very good sort of quick look at sort of the internal state of your body. So it's a thing of governance. We typically do that. Most systems, once they get going, we have to hold them back. The natural tendency is for them to accelerate. So getting back to this, the nuclear membrane barrier that encloses it, it's called nucleoplasm. Don't really care. Yeah, the rough endoplasmic reticulum, ribosomes, the lamina, nuclear lamina is, again, this mesh network. It's kind of made of microfilaments. Most cells have it. It gives it a certain degree of shape. The pores are pore complexes, stuff going in and out. Very, very specific. No, not all pores are created equal. But the big idea from this is DNA is double-stranded. It just can't fit. It's the biggest macromolecule that we have. Where RNA is linear. If as long as it can be very, very long, but it's linear. That way it can get through more easily. It's just, whatever analogy works for you. It's like having a big door or having a little door for, like, one of the cats to scoot in and out. We used to have that. Litter boxes in the basement, the cats would scoot down. The dog couldn't get down there to stack. We've talked to the dog about this. He's got a very diet that's unusual, unique. Does your dog like fruits or vegetables? Oh, well, ours doesn't. He leaves that alone. When you give him something at the end, he goes, oh, it's a vegetable. Broccoli? Yeah. Terrible. So here's the nucleolus or nucleolus. Don't care. They call it the organizer region. Yeah. 1 or 2%. I'll buy that. Involved in, it's sort of a what I call permissive terminal. When you see things like involved in, may be associated with, those are permissive. There's code on these books and PowerPoints for we don't know. But there's enough consistency with this. That's the standard presumption in science these days. Here's the chromatin. Relatively little of that stuff that you saw spread out in the cell is DNA. It's got a lot of proteins, it's got some RNA mixed with it. The fancy term is nucleosomes. I won't ask you about that. That's the DNA. And they say wrapped around the histones. I kind of like it with something that's there and you sort of stick it in and preventing it from becoming, once DNA supercoils, it's ready to go. When you see mitosis under a microscope, those classic slides with when you see actual chromosomes appear with centromeres, it's supercoiled. That's what made it visible. It's condensed upon itself. It's like, in the old days you had an airplane that had a rubber band with a propeller, and the rubber band would get thicker and thicker and sort of develop little kinks in it. That's supercoiled. Keeps folding and folding on itself. So the chromosomes per se are truly, the right word to say it is the condensed genetic material they call chromatin here. And the condensed state helps to protect, yeah, I'll buy that. The threads during, okay, you want to say that? Here's what I mean. So normally this would be all coiled up, really tight together. And by stretching it out, those proteins, to me, keep it in a state, well, does it protect it? Sure. but it prevents it from supercoiling. That's the point I'm trying to make. Here you can see it all coiled up. The proteins have effectively disappeared. In the middle, you have a centromere. That's a bit of protein that permits it to attach to the spindle. And what you see here, when you see this pair, they call them chromatids. So this is a duplicated chromosome already. So you actually have a pair. We call them sister chromatids. And what happens, this is whatever chromosome it is now effectively made two of them by this point. And you're down in metaphase when we get there, when it lines up the most visible of all the phases. It attaches to the spindle by this little nugget of protein called the centromere. Thankfully, we're done with that. I thought it would never end. Which brings us to the last of this sequence of PowerPoints, the cell cycle. That's what today's presentation is about. The cell cycle is that event which is basically about mitosis. For our purposes, we're not looking at meiosis. Mitosis is all about producing exact copies every time, without errors, without structural errors, and particularly without genetic errors, which we have the famous name for, mutations. That's what mitosis is. Our body, and I say it 1,000 times, doesn't repair itself, it replaces itself. It replaces itself with identical cells. Consistent, all the time, continuously. Billions and billions of cells every day are made, okay, to replace others that are no longer functional or have been shed for whatever reason. It's just an array of metabolic activity that's going on. And classically, we have divided this, okay, into mitosis and meiosis. Mitosis is asexual. Basically, it's all about these exact copies, where meiosis, which we study in reproductive physiology, not really part of AP1, is all about things that are diverse. It's very much about creating cells that are really half cells genetically, that have genetic material that's different from the exact genetic material from the parent source that's there. So there's a lot of steps that are in there. There are two giant steps in there that ensure, for all intents and purposes, the fact that they're not going to be identical. That's the interesting thing about them. We see a lot of similarities, but we rarely see things that are identical. I mean, it's different if you're having identical twins. That's a different. Those are genetically identical. Okay. For instance, I always use this. My wife had 5 kids, 4 boys. The 3 older boys, you could throw a blanket over. I mean, they look almost identical to one another. Their general physical appearance, one's a little taller, one's a little shorter, one's a little wider, one's a little skinnier, but you would know they were brothers right away. No question. Only the youngest one picks up genetic material more from my mom's paternal side. My wife, rather, her paternal side. So it's very, very interesting when you look at these things genetically that are there. It's the oddities of it. It doesn't necessarily present itself consistently, though in some families it does. They're there. Remind me one time, I'll show you. I have a recent picture of myself and my sister. Thankfully, she doesn't look a thing like me. See, thank you. I got an occasional smile. It's like if you put 3 of her together, you'd have 1 of me. Really? I couldn't imagine her being 5'8.5" and 200 pounds. That would look really funny. This is a genetic characteristic. I will give you a little family history if you like. My mother's maiden name was Krumm-Holz. Krumholz. Holz in the German means woods. They came from what's called Austria, Poland, from the Krakow area. Unfortunately, very close to a place in history called Auschwitz. But that's the reason why I don't have a lot of family members around. So they emigrated in the late 1890s, the Krumm-Holz family. Holz, generally when you had the family name Holz, they were the men were built like trees, tall and slender. Krumholtz family were built like tree trunks. True story. Her brother, his two boys, my kind of endomorphic physical appearance. Will never be tall and skinny. Moving on. So when I was in school, we dealt with these cycles and all this about the cell cycle, it was very straightforward. They showed you a picture of a cell and they said, there's two phases. There's mitosis and there's interphase. Interphase, mitosis, we see the chromosomes, we see them lining up, we see all the division. That cell is busy. And then we have interphase, and the cell is just there. It's like it's sleeping. Nothing could be further from the truth. Mitosis is a very, very limited amount of the cells. Some cells, maybe as little as 5 to 10%. Okay, the vast majority of the time they're in interphase. That's when the cell does all its activity. In interphase, it's getting ready for mitosis. It's duplicating DNA. It's getting ready to divide. You just can't see it. Okay? And in the meantime, it's doing all its other cellular activities, whether it's processing digestive material, it's moving stuff about in the urinary tract, doesn't matter. Protecting you from infection, they're all doing those things, yet they're still carrying on mitosis and making exact duplicates because effectively cells have a lifespan. So that's the first misconception. It grows and it's getting ready. It's assembling all the building blocks to make a whole new cell. It's actually going to have some of its own DNA. We have a thing called semi-conservative replication. You have two strands of DNA. Remember, we talked about anti-parallel. One's upside down, one's right side up. When they divide to replicate, each cell gets a copy of the old strand and a copy of a new strand. That's called semi-conservative. The difference between that and cloning is like how you typed up a report or you word processor report. You made a copy. Now you took that copy and you copied that. Then you took that copy that you just now made and copied that. You made a copy of a copy of a copy. By the time you make about 20 copies, it starts to fade. Cloning, you lose you begin to destabilize the DNA. Semi-conservative replication keeps a certain degree of freshness, if you will, to the DNA and limits its degradation as you make additional mitotic copies. All right, thank you very much. So the cell prepares for division, carries out its regular activities. Hey, it's busy. In interphase, we don't see it. It's in and this is where the error was in the previous PowerPoint slide, uncondensed chromatin. And it consists of subphases, which include the process of DNA replication. They are well known. You need to know all of these, not for this exam, for the next exam, obviously. G1, gap or growth phase, bless you, other authors, depends what they say, vigorous growth. This is the big phase. Everything is getting prepared. All the building blocks are being assembled while it's carrying on its activity. And indeed, unless everything is assembled completely properly, it will not continue. And you'll see there's a checkpoint, a point of governance that does not allow it to occur. When you see nerve cells, skeletal muscle, cardiac muscle cells, mostly smooth muscle cells, they have reached a point of maturation, okay, what we call differentiation. As the cells have continued to build and build, they've reached a certain point where they will no longer undergo mitosis. This is called G sub 0. The way they've written it there is a little confusing. Not G0 or go, it's G sub 0. And what it means, with a subscript 0 rather, it means that cell will no longer undergo mitosis. It's a good news and bad news thing. The bad news is very clear. Since we don't repair, we replace ourselves. If that cell is damaged, it cannot be repaired. It can't be replaced. It will heal with scar tissue. When you have a heart attack, cardiac muscle cells have died. They are never going to be replaced. They're replaced by scar tissue. Brain cells die. They're never replaced in a stroke. The good news, since they are no longer in mitosis, they cannot develop cancer. Cancer is mitosis gone out of control. And it's like anything else, when the cell cycle loses its controls, it goes faster and faster and faster. It's just like a boulder rolling down a hill. It's just going to accelerate with gravity. And what happens, I don't know about you, but what happens with me when I do something too fast, what do you make more of? Mistakes. And because of that, we lose the controls where we can check those mistakes. When you make enough of those mistakes, eventually you're going to get a cell, an aberrant or irregular cell, that's going to have a potential, as it continues to replicate, to become a cancerous cell. Period. That's what happens. It's mitosis out of control. It has a phase called S phase, or synthetic phase, dedicated to replicating the DNA. The building blocks are out there. All the A's, T's, G's, C's, deoxyriboses, blah, 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 blah. They're all there, but this is when they're assembled. That's when mutations occur. That's what's so vital to understand about S phase. And in G2, it's all getting ready. That's that final check. That's when our cells can chemically see, is the DNA assembled properly? Are all the building blocks in place? Because if they're not, we still have a basic way to say, oh, we're not going to proceed. We'll go back and start over again. We'll destroy that particular cell and move on. Those are all the safeguards. We have so many safeguards to prevent bad cells from occurring. It would be naive to think that we don't have errors in DNA, or we have cells that potentially could be harmful. Why are you and I sitting here, hopefully very, very healthy? Because we have all these safeguards. Bad cells, or potentially bad cells, don't come to light. They're basically destroyed. As long as we don't overdo making them. What overdoes making them? Eating the wrong stuff, not exercising, smoking, drinking, illicit drugs. Pick something bad for you. Getting stressed out when the Steelers are trying to blow an 18-point lead. Never easy. They are the poster children of ugly wins. I go from sitting comfortably in my chair to pacing, screaming, and yelling. I digress. Here it is. You've seen this before, I'll wager. So, and not done to scale. Depending on the average is 10 to 25% is involved in what they call the M phase. Mitosis is not cellular division. This is definitional. It's really important. Mitosis is nuclear division. It strictly involves making a whole new nucleus. You start with one, you got two identical nuclei. Cell division is cytokinesis. They are not equivalent terms. They are an M phase. M phase is not just mitosis. The famous prophase, metaphase, anaphase, and telophase or telophase are mitosis. Starting somewhere typically toward the latter part of anaphase, we end up with cytokinesis. That's when this cleft or cleavage furrow forms and eventually the cells develop. Understand that. These are the critical players, the checkpoints, G1 and G2 or G2M. This is known famously as the restriction point. If everything is not assembled sufficiently or there are inherent mistakes, that cell will destroy itself. What happens is a series of waste products that you'll come to see shortly that are called cyclins, C-Y-C-L-I-N-S, are formed. And they join with an enzyme, and effectively when they join sufficiently with this enzyme, they release this checkpoint. Now you got the boulder at the top of the hill, and you've got something, and you release it, it starts to move. That's what I mean by governance. We call it a restriction point. It restricts further activity. Assuming it has all its quote-unquote ducks in a row, to use a common cliche. Now we go into growth and DNA synthesis. Yes, it's growing some more, but the big player here is the assembly of DNA, all the A's, T's, G's, C's, etc., etc. That's when mutations occur. And then it continues on for these final preparations in G sub 2. Effectively, this is looking for, metaphorically speaking, for cellular mechanisms to identify, are there errors in DNA? Are there errors in the assembly of all the materials necessary for replication? And if so, if everything is in order, then yet another series of waste products, similarly called cyclins will be there, and they will allow this restriction or checkpoint to be passed, and this blocker will be sort of biochemically removed and allow mitosis to proceed. And we just start all over again. And every type of cell, your gut cells are doing this the most rapidly, replacing all of them every 3 to 6 days. Some cells, skin cells, the lifespan of a skin cell is about 25 to 30 days, from keratinocyte and stratum basal all the way until it's shed. It kind of gives you an idea of metabolic rates. You know, somebody has psoriasis, by and large, it's an autoimmune disorder that makes those cells mature too rapidly. So there's all cells have how rapid they're supposed to go. We're not doing this. Be thankful. Replication forks and replication bubbles, depending on who would teach you A and P or who would teach you micro, they will sometimes go into this. This is classic understanding it. I don't even go understand it with my advanced students. They have a course in cell and molecular biology and a course in genetics that specifically deals with this. I don't feel that the students in this level have a need to know this comparatively, depending on where you're going. So effectively, when they divide, they call it a fork. The area that surrounds that divide and it moves is called a bubble. Okay, and then it just starts going from there. There's all sorts of different. I wrote down one of these chemicals, this DNA polymerase. That's you have to know. DNA polymerase with the Roman numeral 3 is very important. The reason we don't get a lot of mutations is this. You remember earlier when we were talking about DNA, I said the A's and the T's and the B's and the C's. If there's an error, you were G, shouldn't be here. In bacteria, it would replace one of them without knowing, so to speak, which one is the correct one. It's likely to replace the good strand with mutated. DNA polymerase 3 is a proofreading mechanism that not only can identify the mutation, but it can identify which strand is inappropriate. It may replace that strand. That's a very elaborate proof. So the more advanced your cells, the more advanced the proofreading we have. So it's not that this isn't there, but you're not going to have to look at replicases, polymerases, and ligases. Okay? Be thankful. I mean, it's a lot of extra nomenclature. For bio majors and pre-med majors, it's important. So the end result is important to us. 2 identical daughter DNA molecules. And again, this semi-conservative replication, as I explained, 1 strand of the original DNA and 1 duplicated strand. Here's the fork. Here are the bubbles. This bubble is like the whole area. And they're moving. And if you want to get into it in detail, I mean, there's something called a leading strand, where it's going in the right direction, and a lagging strand. And this makes little fragments that you have to eventually sew together with something. When you ligate something, you tie it together. The ligature in surgery is you'll have a bleeding tiny blood vessels. You'll do a tie around it. Nowadays, you basically chlorize them. You ligate the vessels. If you like crime shows and they show somebody whose wrists were bound post-mortem, they'll say, oh, they have ligature marks. See, you learn something new every day. Favorite crime show is back for another season. On BBC, it's called Silent Witness. It's great. What's the first episode of the new season last night? Our British friends have an absolute addiction to crime shows, all those Agatha Christie novels and stuff like that. Life looks good. Not another one. Not doing it. So most cells need to replicate continuously is a good word. It's just how frequently continuously means. Skeletal, cardiac, and nerve cells do not divide. Scar tissue, that whole thing before. Understand that we can't avoid it sometimes. A lot of times growth is hard to predict. will operate on somebody, will make scar tissue. Sometimes it's overabundant. It's very hard to predict how things will heal or not heal. There's so many variables that are involved. I cited the other class, a virus that many people get potentially could be cancerous. For most of us, it's not. It causes an excessive amount of growth. Even it's actually a benign tumor. It's a wart. All warts are made from something called HPV. You've heard of human papillomavirus. There are certain strains of it that, unfortunately, when they're associated with cervical or penile tissue, they frequently become cancerous and frequently are deadly. No two ways about it. What it does is it creates a rapid cell cycle, and you get abundance in typical warts of benign, just a whole bunch of normal cells that are typically relatively easy to go away by themselves or easily dealt with by a variety of methods, topical treatments, surgical treatments. I have treated thousands by all of those methods. The question is location and stuff that's done to them. Years ago, these treat warts with x-rays. You treat something that was benign, something that was malignant. But just to give you an idea, so I have a fair amount of experience in dealing with these things. So M, or mitotic phase, is in which division occurs, consists of the two events, mitosis and cytokinesis, nuclear division, cellular division. The control is the pivotal piece, back to the same cycle that I described. Here are the events. But the only part that you have to know, really, about this, however you measure, I use the little mnemonic ANT, prophase, metaphase, anaphase, telophase. You're welcome to use it for four things occur in prophase. In telophase, they're reversed. Metaphase is the easiest. They line up in the middle. They begin for metaphase. And anaphase is the shortest, and they separate. And this is all, it's the concept. The chromosomes appear. They supercoil. So those histone proteins disengage and it begins to coil and it becomes visible. The nucleolus disappears. It's a visible thing under a microscope. The nucleolus is still there. It's just separated enough that we don't see this entity under the microscope. It's changed its conformation. The same thing with a nuclear membrane. It's a membrane. By definition, it can open and close and separate, reform. So it appears to disappear. The nucleolus appears to disappear. And lastly, the spindle, which is made of microtubules, appears. Again, you saw that on the animation. Those little microtubule subunits begin to assemble. Once they assemble, we can see them. When they disassemble, we can't. So we've described these events by what we can see and what we cannot see. It doesn't mean that the materials are still there. They're just not in a visible way when we look at something microscopically. But we use those as landmarks. Again, nucleolus disappears. Nuclear membrane disappears. The spindle appears. Chromosomes now condense or supercoil and appear. Those are the four events. You're going to see even in this particular slide set, they talk about early and late. I don't care. It's just prophase. Metaphase in the middle, anaphase, they split, telophase, reversal prophase. So here's early prophase. Yeah, we start to see the chromosomes. They're already duplicated. You have the double pairs. The centromere is this knob of protein in the middle that's there that is going to eventually attach to the spindle. You begin to see that, the microtubules. Remember, you have those two oblong centrioles. They move around and start producing the spindle. And then eventually the nuclear envelope breaks up and all the things with kinetochores. Don't care. Just those four things. So here's interphase. I don't think it's a great illustration because if I can see chromosomes, I'm already on my way to prophase. This better illustration. You're beginning to see what they call the asters. The centrioles are dividing and moving to the sides. Here you can see the chromosomes already duplicated. And now they have, so actually, when you look closely at it, there's actually 4 strands rather than just 2, Not a big deal for us at this point in time. And in the middle there, you begin to see that little nugget of protein we call the centromere. Now, as you get later in prophase, now you're seeing the spindle, the nucleolus is long gone, nuclear membrane is long gone, and they're beginning to line up. When they line up along what we call the metaphase plate, we have the easiest one to identify. Microscopically, they're lined up in the middle, and then now they're just going to separate. It's basically like a pulley system, and the centromeres are where you're going to see the sister chromatids, these pairs of chromosomes now begin to separate. They've already duplicated, so there's 2 on one, 2 on the other, is really 4 held together and apart. Anaphase, centromeres of the chromosomes split. Each sister chromatid, actually a pair of sister chromatids, is pulled across to the original side. Voila. This happens relatively rapidly, and then everything begins to reverse. Chromosome movement stops. Now they're at the opposite ends. It begins to uncoil or unsupercoil, and chromatin forms with the association of the histone proteins, their membranes, nucleoli, spindles, blah, 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 blah. And sometime in that, we begin to develop what they call the cleavage furrow. That's the cytokinesis, that's cellular division. The best analogy is like, I love it, it's a balloon puppet. You ever do that and you take a balloon and you start to just twist it? It develops a furrow. It's really actin filaments on the outside are doing that. And if you keep doing it, theoretically, you'll be able to pull it apart. Whenever I tried to do that, it's just a plumbers at our house this morning. I saw the number. It's like I'm not having a good morning. So here's that furrow that's there. And they go through all the steps. You can look at this if you want. And again, the big thing for our purposes are these controls. Oh, good. Teeth marks. Pardon me for a second. Moving on. So here are some of the things that control it. Sorry for having to answer that. So these are the signals. The whole thing, surface to volume ratios. Very, very important. Think of this room, surface to volume. Think of the relationship between the 4 walls, the ceiling, and the floor. That's surface area of a cell. For a cell to function, things have to be able to get to the center of the cell relatively easily. And by the same token, you have to be able to spit them out by way of the ceiling, the floor, or the walls. The volume, as it increases much rapidly, as a cell grows, okay, remember the volume is something, is a cubic measurement, so it's a measurement of width and length and height. So if the height of this room would magically be able to expand, we would have increasing volume and the surface area would not change as rapidly. So that's why, even though our cells are smaller, proportionally smaller than an elephant or a whale's, it's not proportional. Their cells are a little bit bigger, but they have to have the right surface area to volume ratio in order to function. So there's a lot of things that influence those go signals. When the cell starts to get too large, okay, that it couldn't do business, it separates or replicates. Typically, cells divide by something called contact inhibition. When we do cells in culture in a laboratory in a beaker, they lay on the top. They don't heap up and go this way or push down and go that way. They don't form a massive lump of cells. They're just sort of like a linear, sort of a flat layer. That's because of contact inhibition. Once all the cells are cuddled against each other, they lose the impetus to grow. That cancer cells lose those controls. They just keep growing. Particularly contact inhibition. They become very invasive, irregularly. They attack healthy cells. They utilize them for nutrients. These are the chemicals, the cyclins, that control those checkpoints. Cyclins, they call them regulatory proteins. Think of them as waste products. I do. And they bond with a particular kinase, an enzyme, and they form this complex of cyclin with an enzyme. And what that does is basically unlocks the checkpoint and allows it to proceed. G1 goes to M all the way to G2 into the next checkpoint. If it clears the G2 checkpoint, then it can go on to mitosis and start the cycle all over again. And the cyclins are destroyed every time there's mitosis. We start all over. They're not lingering around. And so those are the key events. The G1 is the most important of all those checkpoints. The G2 and the M are considered two checkpoints, even though they're very, very close to one another. The second set of checkpoints that are there effectively. Okay, that G1, if the materials are not there in a sufficient amount, if we don't have a sufficient amount of building blocks for DNA, building blocks for membrane, then the cell just can't be made. And so that's really a substantial control. The one that's sort of on the lookout for mutations is that second checkpoint, the G2M checkpoint. Okay, and again, what happens, a lot of these PEDs, they don't all necessarily go to G sub-0, where they stop replicating. Some of them undergo programmed cell death. We call that apoptosis. I do not think we're going to get to that today. That's probably a little bit. Remember, on Friday, I won't be here, and it's really just summary stuff. I'll summarize what's left to talk about with the cell, and I'll give a basic introduction to bones, and then we'll get in sort of into the meat of bones. Even though you may have had some of this stuff, we really want to look at the ultrastructure of the bone itself, and eventually get into how it's assembled, disassembled, talk about fractures, diseases, controls, etc. Okay. And really, that's what you have to understand. We're not going to do this. This whole portion of the remaining of this, if you want to look through it, whether in a chapter, or if you want to look through it, just buzz through a PowerPoint slide, indeed do so. So we all know DNA is the master blueprint. It talks about, it's a letter of, it's basically a 3-letter language. And it goes from something we call these triplets that become things called codons and anticodons. And really, the segments that have this are referred to as genes. Again, a lot of the part that makes the proteins is really, to me, the gene is the whole segment. It has a lot of regulatory material, and it has the area that specifically codes to make the proteins. So again, you're going to look at these triplet codes, and then you can go on. And I don't do any of this stuff, because I feel it's more than we need. Our messenger RNA is huge. We chop off regulatory pieces. What leaves the nucleus is effectively introns or insertion sequences. If there was a mutation on an intron, it would not affect the gene because it is removed. And basically what we do is we splice together what are called exons, so that big long piece of messenger RNA is shortened from pre-messenger RNA to the actual messenger RNA. It's called messenger RNA processing. It's one of the safeguards that limits the likelihood in our cells compared to, let's say, a bacterial cell. Bacteria mutate all the time. Viruses mutate all the time. They don't have the genetic safeguards. I mean, right now, RNA is inherently unstable, and viruses, think the flu, think COVID. Bacteria are constantly mutating. That's the biggest problem we face. For years, we had Staphylococcus aureus as being the big culprit. Now, 70 to 90% of all Staphylococcus aureus is something called MRSA, which you'll all learn about, called methicillin-resistant Staphylococcus aureus. That's what makes it so much a problem. We can't treat it very easily. We're running out of antibiotics. Because of what? It mutates. It doesn't have DNA polymerase 3. It doesn't have all the things, and I go over this chapter and verse in the genetics area of micro. So this is just a little introduction. I'm not going to ask you a single question about exons or introns. This, if you want to look through it, doesn't help me. Okay, RNA, messenger, ribosomal, and transfer. Very straightforward. Those are the three types. This is carrying the message to the ribosomes. This is the platform. It's made of ribosomal RNA and protein. Transfer RNA works with this three-letter thing we call codons and anticodons, and make sure the right amino acids are brought in the right sequence. And so effectively, you want to summarize it in the interest of time. Transcription is DNA information coded into messenger RNA. Translation is that messenger RNA eventually made into polypeptides, or if they're big enough, proteins. Here it is. This is the pre-messenger RNA. This is how long it is. Look what's actually leaving. So there's lots of different, you're not going to have anything about the transcription factors or RNA polymerase or any of that stuff. It's really important stuff medically and biologically to understand the process. Nobel Prize within the last 20 years was all about kind of RNA polymerase. Why are we studying it? Because if we can impact, if we can limit bad things being produced, we can limit cancers. That's where the research is. It's on a molecular level. Either enhance DNA polymerase to eliminate more of them or block them from starting. So understanding RNA polymerase. I listened to the Nobel Prize lecture because they all give a lecture to the assembled people. My head's pretty high. This is way above my pay grade. And I'm not going to do anything about IEs and Ts. This is sort of like the assembly line for making proteins. You don't want any of that stuff. Trust me. None of the events. Someone, not me, might throw one of these at you to understand this is all in the language of messenger RNA. This is what we call the genetic code. A lot of duplication and all the different roles that are there. Like I said, I'm not going to do it. You don't have to thank me. I don't even enjoy teaching it. Polyribosomes are kind of interesting. Down the road, when you see about how we fight an infection with antibodies, why can we make millions of molecules in a matter of minutes and flood our bodies with it? Once that ramps up to speed. Here's basically the strand of messenger RNA to make those antibodies. We hook it up to multiple ribosomes, so and it's just producing them massively and just shifting down the line. So we're producing massive amounts on one strand all at the same time. That's what cells do that are called plasma cells that you'll learn about. And so I just thought I would throw that out for some just giving you some sort of idea of what's going on. I'm not going to do anything with SRPs or clipping them and any of that crap. No. We're going to stop here with the algorithm. I do want to comment about this, the apoptosis and the autophagy. And again, I'll see everybody as soon as you get here. Start next time. Thank you.